It's like it's such a waste of time. Everything I do in lab. Such a waste of time. You don't learn anything.
I mean, it was interesting. It was like kind of stuff that we already talked about in the summer.
But like it won't help. No.
At least because right now I'm not really.
Oh, I agree. Yeah. I think I know you're almost. Yeah.
There's even fewer when we got here. That's true.
Okay, let's get started.
We should switch to the rating. Exactly. That's what you want to
Yeah, I'm saying like we're also.
Yeah. I saw Alpha. Oh yeah.
Okay, it seems we're still in the practice session. Okay, all right, thanks, Mike. Okay. I'm pro. Okay. All right. Welcome everyone to our BME seminar series hosted by the Department of Biomedical Engineering at Columbia University. My name is Josh Jacobs, and I'm an associate professor of biomedical engineering here at Columbia. Today, we continue to offer this lecture in a hybrid format with both a virtual and a live audience. Our speaker will present for 40 or 45 minutes, and we'll leave 15 minutes or so at the end for question and answer. For our virtual audience, throughout the presentation, please use the Q&A feature at the bottom of the screen to submit your questions, not the chat function. We will try to answer as many questions as possible during the Q&A session at the end. For our live audience, of course, you have an opportunity to answer questions, ask your questions interactively with us.
I got raised.
Today we are excited to welcome Professor Mallidi. Dr. Mallidi received her MS and PhD degrees in biomedical engineering from the University of Texas at Austin. Dr. Mallidi's master's thesis involves custom-designed ultrasound-based multimodality imaging systems, and her doctoral work for the utility of surface plasma resonance properties of spherical gold nanoparticles for early cancer detection and probing of nanomolecular interactions. After graduation, she joined the Wellman Center for Photomedicine at MGH Hospital as an NRSA postdoctoral fellow. She's received several Young Investigator Awards from national and international conferences, and she currently directs the Integrated Biofunctional Imaging and Therapeutics Lab at the Department of Biomedical Engineering at Tufts University. Her research interests are in the field of ultrasound imaging, photoacoustic imaging, image-guided therapeutics, photodynamic therapy, and combination treatments. Welcome, Dr. Lee.
Thank you so much, and thank you to the organizers for inviting me here. Can everybody hear me okay? Yeah. All right, okay. I'll try to speak louder also because for the virtual audience, they need my voice to be a little bit louder. All right, so we're all set. Okay, I'm going to remove my mask so that you can speak a little bit more clearly. Hello, everyone. I will be presenting today to you 3 stories about ultrasound and photoacoustic imaging and how we are using that technology to personalize treatment strategies. So specifically, there's some nanotechnology involved and of course, ultrasound-guided photoacoustic imaging. But the 3 stories are, how can we predict tumor recurrence using this multi-parametric imaging modality? How can we plan combination treatments? And how can we monitor nanotherapeutics? So if you're delivering drugs using nanotechnology, using ultrasound photoacoustic imaging. So the 3 main topics that you will see in my talk is tumor hypoxia, and I assume all of you know what tumor hypoxia is. When tumors grow to are large, there's inadequate supply of blood vessels and lack of oxygen, right? So there's a lot of tumor hypoxia focus in today's talk, and I'll be talking about photoacoustic imaging and photodynamic therapy. So I'll start with introducing what photodynamic therapy is, assuming that that's something new for the audience here, and then talk about photoacoustic imaging, and then tie it all up with tumor hypoxia. So what is photodynamic therapy. Basically, it's a light-based therapy and has two parts. The photosensitizer, which is a light-activated drug, and you shine light once the photosensitizer is delivered to a tumor. You shine light on the tumor. The light at a specific wavelength excites the photosensitizer, and in the excited state, all right, okay, great. And in the excited state, the photosensitizer reacts with oxygen to react to oxygen species. And these reactive oxygen species can destroy the tumor cells, and therefore you get the destruction of the tumor. So photodynamic therapy has been in field and in use for a long time, but really did not make its impact into clinic as we would have hoped. I'm going to show you a few examples where it worked tremendously well, and also talk about why we need photoacoustic imaging to guide this therapy. So this is an example from Dr. Meryl Beal in Mayo Clinic. This is a patient who has recurrent squamous cell carcinoma, essentially went through surgical resection and also radiotherapy, but then had recurrence, went through surgical resection and chemotherapy again. And then a year later, there was a recurrence to the upper neck, as you can see here. So almost all of the therapies did not work in this particular patient. So this patient was treated by Dr. Beal with PDT. So here you can see actually a photograph from the clinical room, the fibers being delivered specifically localized to that region of the tumor. And this patient is cancer-free after, you can see that the region is healed well and is still smoking. I'll show you another example, a study where that I was very closely involved. In India, oral cancer is a big problem, right? 1 in 6 patients dies of oral cancer, and that's because of the habit of chewing tobacco. A lot of people chew tobacco and have these lesions, these early lesions. So this is a T1, stage 1 cancer lesion in the oral cavity of the patient. This is a photograph of the lesion before treatment. 1 week post-treatment. So again, this is very good because you're doing localized therapy. You're delivering light and the photosensitizer at that location where the lesions are, unlike having radio or chemotherapy, which is much more systemically toxic, right? And you can actually see 8 months post-treatment, the follow-up, right? There is very good healing of the mucosa. The patient is cancer negative and everything is good. So good responses, and you're seeing such good healing properties and healing of the mucosa. What is happening with PDT that it did not make it into the mainstream yet? And there are 2 main problems. One is the delivery of the photosensitizer, right? How can we specifically deliver the photosensitizer only to the tumor and not anywhere else in the body? Because if it goes elsewhere and you shine light, even your normal tissue might be impacted because of PDT. So the specificity of the photosensitizer to the tumor is very important. And another major thing is this oxygen term that you're seeing here. If there was no oxygen in the vicinity of this photosensitizer in its excited state, right, it's just going to remain there and relax probably, but there's no oxygen for it to generate these reactive oxygen species. So oxygen is very important. So if the tumors were hypoxic to begin with, PDT is ineffective. And this is not only true for PDT, even for radiotherapy, tumors that are hypoxic, right, the response is not that great, and for a lot of chemotherapies as well. So impact of tumor oxygenation on the response is very important. Tumors that are non-hypoxic, you can actually see patients have a longer fraction of survival in non-hypoxic tumors. In hypoxic tumors, the survival is very low. And also, if you look at oxygen saturation, blood oxygen saturation, we are very familiar with this term right now because of COVID. We'd use the pulse oximeter to measure our StO2. The same thing you can also, measure in tumors as well. And you can actually see that tumors that have low oxygen saturation did not have complete response versus tumors that are highly oxygenated had very good response to treatments. So overall, looking at the tumor oxygenation content and the blood vessels that are feeding these tumors becomes very, very important when we are trying to personalize this photodynamic therapy. So oxygen is very key, and whether the photosensitizer is being delivered to the tumor or not becomes very key as well. So why blood vessels? They feed the tumors. And today I'll be talking a lot about this particular term called StO2 or oxygen saturation, which is nothing but a ratio of your oxygenated hemoglobin in your blood to the total hemoglobin, right? So we are talking about tumor hypoxia, and we now know that it is very important to understand whether a tumor is hypoxic or not, to understand whether, or to gauge whether your treatments are effective or not. So what are the modalities that are out there that can measure tumor hypoxia? So several modalities do that. PET, if you inject something called F-meso, you can understand the tumor hypoxia, but then, these modalities use exogenous contrast agents. The non-ionizing imaging modalities, MRI, you could do bold MRI as well. But here I'm focusing on ultrasound and optical imaging more, which is what is photoacoustic imaging. So we want to take the salient features of ultrasound. So now I'm moving from photodynamics and introducing you the photoacoustics. So ultrasound is cost effective, it is mobile, it's real time, has good penetration depth. So, and then optical imaging modalities can provide molecular information of the hemoglobin. So if you oxy and deoxyhemoglobin have different optical properties. So you can use optical imaging to get the molecular information about hemoglobin. So you combine both, you take the optical imaging, it's very good with functional molecular information, and you have the ultrasound imaging that is frequency dependent, but has very good penetration depth, has good resolution, but cannot give you, oxygen saturation values. So you combine those two to get a photoacoustic imaging modality. As the name sounds, photo, light is generating sound. How many of you have heard of this modality? Yes? No? Okay, great. Yeah, so it's light is generating sound, and you use the same ultrasound system that is used in clinics worldwide to actually detect the same photoacoustic signal. So now, is any light generating sound, or do you need a specific light, pulse or energy to generate sound? So general tube lights or any fluorescent light can't generate a photoacoustic signal. You need a specific nanosecond pulse laser to generate a photoacoustic signal, and this nanosecond pulse laser satisfies the thermal stress confinement condition. All right, and in that, you're confining the stress, you're confining the thermal response, and because of that, there's a sudden boom effect, right? And that generates the sound that is detected by the transducer. So I'm going to show you here an example. So assume you have the surrounding tissue that is scattering, is not absorbing light. And assume that this gray circle here is a blood vessel that has blood and hemoglobin, obviously. So a transducer is what is used in ultrasound imaging. You transmit sound and receive sound to do regular ultrasound imaging. In photoacoustics, the ultrasound transducer is surrounded by the optical fibers. So you excite this particular tissue with a nanosecond pulse laser. And that laser energy is absorbed by the blood vessel and undergoes a sudden thermoelastic expansion and contraction and a local increase in temperature. It is in the millikelvin range and generates a pressure wave. The pressure wave is then detected by the transducer. So the scattering tissue, which is on the surrounding, is not absorbing this laser energy, it's just scattering all of the light. So you get a photoacoustic response only where there is absorption of the laser, right? So if you generate a photoacoustic image of a representative image here, the surrounding where you don't have any photoacoustic signals, it's a scattering medium, and your absorber or your blood vessel, in this case, the inclusion, where you have a lot of absorption, is where you see a photoacoustic signal. So if you think of it, right, the pressure that's generated is dependent upon the wavelength. So if you're using 532 nanometers versus 680 nanometers, different wavelengths have different penetration depth, and our optical properties of the tissue are also wavelength dependent, right? And also with respect to the depth. So unlike some imaging modalities where we don't get depth information, because photoacoustics is ultrasound based, you also get information based on the depth. So you can actually look in Z, here Z is represented by the depth in the tissue. So the amount of energy that you input, which is your fluence, and the optical properties of your tissue will determine what a photoacoustic signal strength is. So if I'm trying to image oxygen saturation, right, I can look at deoxy and oxyhemoglobin and tease out the oxygen saturation in depth in 3D volume for these tumors. So another example before we actually go into tumor work is, again, this is a tissue mimicking material. You sort of see the black absorber, which can absorb the light. Ultrasound, which is the same transducer that we're using for photoacoustic imaging, transmit sound, receive sound back, can give you the structure. So we are seeing the structure or the shape of this tissue mimicking material with maybe the inclusion. You can sort of say where the boundaries are. It's not that clear here, but you can see in photoacoustics, now you're acquiring the photoacoustic image at the exact plane that you acquired the ultrasound image at, right? So since the surrounding tissue is all white, it's scattering, and you have this graphite inclusion in the middle that is absorbing the light. So you see the photoacoustic signal just from the absorbing material. And because they're acquired by the same transducer, you actually get spatially co-registered images. So ultrasound is giving you the space information, and photoacoustics will give you the molecular, functional information of the blood vessels. So if we are to now compare, the imaging modalities, particularly optical imaging modalities, I know, a lot of folks here and students who work with microscopes. So the standard microscopes or OCT have very good resolution, but the penetration depth is limited. On the other hand, we have diffuse optic tomography or NIRS, where you have very good penetration depth, but the resolution is not that great, right? So photoacoustics sort of falls in this mesoscale, where you have decent penetration depth and a decent resolution, and that resolution is determined by your transducer frequency. So higher the frequency, the better the resolution. So in photoacoustics, again, to rephrase, we can do multi-wavelengths. So you have oxy and deoxy hemoglobin that have different different optical absorption properties. Photoacoustic signal is dependent upon that wavelength and also depth. And if you're imaging at multiple wavelengths, right, we have two unknowns here, oxyhemoglobin and deoxyhemoglobin. You image at 2 wavelengths, you could also image at more wavelengths. You can tease out what the oxygen saturation of a particular tissue is. And it's not just one single point measurement, you can get this oxygen saturation for the whole tissue in 3D. So summarizing again, photoacoustic imaging will give you oxygen saturation maps because of the absorption coefficient dependence of the photoacoustic signal, and it can be done simultaneously with ultrasound. So I'm going to start showing some in vivo data, and then we'll go back to the photodynamic therapy and how we use photoacoustics with the photodynamic therapy and trying to evaluate or gauge the efficacy in photodynamic therapy. So here you're seeing a nude mouse with a subcutaneous tumor. So X and Y is the plane of the slide, and Z is into the depth or into the skin of the mouse. And the image that you're seeing here is an ultrasound image. The tumor was imaged at a particular cross section shown by this black line here, and the image that you're seeing is now in X and Z, so into the depth, right? So the red region here is the tumor. The skin appears to be very hyper-echoic, right? You see a lot of signal there. And since we can do ultrasound and photoacoustic imaging simultaneously at the same cross-section, you also can get the oxygen saturation map. So this is a post-processed image. So we imaged at multiple wavelengths, did a lot of math, post-processed, and then put it in a pseudo color where 100% oxygen saturation is represented in red, and blue-black is represented in the hypoxic regions or low oxygen saturation images. So this is great. So we are able to get this information, but then we have to validate that with histology. Right. So here is those images again. We did H&E, which is very typical of the structure that can give you the structural information. But to really understand hypoxia and blood vessels, we did CD31 and used a stain called pimenidazole that specifically stains for hypoxia. I would want you all to look at these two images specifically. Right. So wherever there is high oxygenation content, that is red areas, hypoxia should be low. So you can appreciate all of this bottom part of this particular tumor was highly oxygenated. A lot of blood vessels were feeding this tumor from underneath right here. And you can actually see there is no hypoxia stain there because that area is very highly oxygenated. Also towards the left side of the image here, and you can also see this region being highly oxygenated as well. The center part, or towards the right-hand side of the image, you see that the image is highly hypoxic, and that's exactly where the hypoxia stain also holds, right? So there seems to be a very good spatial representation from the photoacoustic image, and it can show the hypoxia that we validated that with the hypoxia stain here. So stopping here, I showed you what photoacoustic imaging is, how it can measure oxygen saturation. We talked about photodynamic therapy, and I told you what photodynamic therapy is, and the problem with photodynamic therapy efficacy is the presence of oxygen, right? So 3 questions that we'll try to answer with photoacoustic image guided photodynamic therapy is, first one is, can we predict tumor recurrence, right? And in that, there are 2 more questions that we want to answer. So without any exogenous contrast, no exogenous contrast agents, can we stratify the patients at a pre-treatment stage into responders and non-responders of treatment? And once we do that, can we gauge the heterogeneity and predict the treatment response, or the recurrence early in the process, right? Without having to wait for a long term to understand that whether a tumor will recur or not. Can we predict that at a much early time point? Because that will be key, right? If a treatment is not working, you would want to treat the tumor in a different way, right? So understanding whether the treatment is working or not in a particular tumor is very key. So to do that, we did a mouse study, and this is again very representative of a lot of therapies. When you have a bunch of mice, right? In this case, there were 40 mice. Some of them were non-responders. As you can see here, the tumor volume just kept increasing, shown in green. And we have complete responders. That means that the PDT was effective, and the tumors went through complete remission, and the mice are doing great. So we'll first compare these 2 categories, look at how photoacoustic images change in the non-responders compared to the complete responders. So I'll be showing you the pseudo-colored images again here. So this is an example from a mouse in the complete responders group. So this is before the photodynamic therapy treatment. The ultrasound image shown in grayscale is on the background, and we overlaid that with the photoacoustic image on the top. So again, very representative of the tumors. The vessel density is heterogeneous. The top region here is highly oxygenated, some hypoxic, low oxygenation areas in the tumor. And 24 hours post-PDT in this complete responders, all of the blood vessels were shut down. So the tumor went into a hypoxic mode, all the blood vessels are shut off. So, that worked for this particular tumor, and we see a complete response. In the non-responders, you can appreciate there was no change in oxygen saturation pre or post-treatment, right? So in a non-responder, we could see that in 24 hours post-treatment, right, that they were completely non-responsive. Now, going back to the tumor volume, you only can see a statistically significant difference between the non-responders and responders 14 days after you start the treatment, right? But with photoacoustics, we were able to tell within 24 hours that a particular mouse will be responding to the treatment or not. And we did a lot of statistics, but then we're just showing the bar graphs here that in complete responders, there's a drastic change in oxygen saturation. In non-responders, there's not much change. Now it gets interesting here, the story gets interesting. And we had more mice included in this group. And some of them actually fool us in trying to be responders. So especially focus on these red tumors that fall under this red line. So initially they showed that, they appear that they're responding to the treatment, but then they start to regrow, and they regrow very aggressively. Now it's very important to understand these tumors also and identify this recurrence much early on, so that maybe you could treat them, more effectively early on. Probably the dose was not enough, that the whole tumor was not treated completely, so there is this recurrence. Often not in clinic too, recurrence is a major problem, and it is also very hard to treat tumors that first became resistant to the treatment that you're giving and then recur later. So can we predict this tumor recurrence? So which the mice that fall under this red line using photoacoustics. So to do this, just looking at a 2D image is not enough, right? Because tumors are heterogeneous. Why is this recurrence happening? Is probably because of that heterogeneity. Some of them responded, some of them did not. So to really understand the heterogeneity, we need to look at the tumor oxygen saturation in 3D. And that's exactly what we did. And we computed something called a prediction map, and I'll walk you through that right now. So we did a lot of 2D scans of both ultrasound and photoacoustic images. And from these 2D scans, I think the video is stuck here. We can generate a 3D volume, tumor volume. Ultrasound will give you tumor volume. Photoacoustics is giving you this oxygen saturation map now in 3D. And you remember our data from before between complete responders and non-responders? We really showed that in complete responders, oxygen saturation decreased significantly. So we just put a simple basic, threshold algorithm of this 3-dimensional data and showed, it said if a region in that tumor volume reduced oxygen saturation, paint that in green and say that the region is treated. If the oxygen saturation in that particular volume did not change, right, paint that region in red and call it a non-responsive region. So you're going from this image of a 3D photoacoustic oxygenation map to something like a green and red sort of a map that we call the prediction map. And I'm going to show you examples of a few tumors that were in the complete responders and non-responders and this recurring group. So 3 panels here. These are photographs of the mice before the photodynamic therapy PDT treatment. Post-treatment photographs are shown on the right-hand side. And then our prediction maps, which are obtained at 24 hours post-PDT, are shown in the center. So the first case here is from a non-responder mice. Obviously, the tumor grew, the tumor volume is higher, and our prediction map predicted that this tumor was treated but was non-responsive. The oxygen saturation did not change much in these tumors. So yeah, we can stratify, that's great, but this is where it gets really interesting. These 2 mice were injected with the same cell line, same number of the cells were treated with the same PDT dose. Everything was as experimentally controlled as possible. But what you see here is 3 days post-treatment, both these tumors start showing necrosis. And if you're just looking at the photographs, one would be excited, like saying, yay, these tumors are on their way to getting treated. These mice are going to be fine. But 23 days later, that is almost 3 weeks later, this mouse, the tumor is completely gone, okay? And there's very good healing of the skin. But in this particular mouse, you can actually see a tumor starting to regrow, right? And this is, again, 3 weeks later, right, when we start to see the recurrence. Using a prediction map, we were able to, at 24 hours post-treatment, actually say that, all of the blood vessels in this middle tumor, the responder tumor, were all shut down, and the tumor became completely hypoxic. All the vessels were shut down, which led to, complete remission. But in the case of regrowth, there was a huge blood vessel in this top region, which the dose just was not sufficient enough to kill that, to shut down that blood vessel. And that's exactly the region where the recurrence happens. And after we detect this, you can see in 1 week, from 23 days to just 30 days, this becomes a really big, huge, drastic tumor, right? So if we had known at early time point before, if we had this prediction model, we could have gone back and actually treated that region, which was non-responsive, and probably not seen this situation. Right, so we were able to identify at 24 hours post-treatment the recurrence that's happening in these mice models. So, when I presented these results to my colleagues, they did not believe me. It's like, oh, you can't predict at 24 hours. That's not possible. So what they cleverly did is, this is a mouse, right? So a photograph of the before treatment picture. They actually covered, and I was blinded to this study, right? They covered half of the tumor with a black cloth. So remember, I was talking about photodynamic therapy, how light is important along with the photosensitizers. So if there is no light, PDT is not going to happen. So if in this case, if they're treating this light illumination all over, the treatment is only going to happen in the top half of the tumor, and the bottom half is not going to be treated. So this is almost like a self-control, if you will, right? And as expected, 1 day post-treatment, you can actually see the top part, has this photothermal damage. The bottom part was, as is, not much damage seen there. And what we get from the prediction map is actually 3 regions. The top region, which was treated, the middle part, which was not, and then we also noticed that the bottom part was not treated, right? So you can actually see this bottom part is not treated because of the black cloth. The light did not get through there for the photodynamic therapy action to happen. But then there is the other region, it's treated as non-responsive, probably, again, like in the other case, had a bigger blood vessel that was just not shut down for the dose, light dose that we delivered here. And 7 days post, you can actually see these 2 regions combining, and then regrowth happening from the top part, and then the whole tumor just becomes aggressive all over again. We validated this with several mice. On the x-axis that you see here is the axial length that we measure of the regrowth region 24 hours post therapy, to caliper measurements, which is much more gold standard in mice work, in preclinical work, and try to measure the same thing, and we do see a very good correlation. So what we're looking at here is change in oxygen saturation post therapy. So, and we also, have a hunch that probably the bigger blood vessels, were not shut down. So what we wanted to do is, what is happening pre-treatment? What is the status of this tumor before actually treating? And can we predict from the pre-treatment and the post-treatment changes how a tumor is going to respond? And this is very preliminary data, and I'm going to show you on a graph here. On the x-axis is the pre-PDT, or the pre-treatment oxygen saturation of the tumors. And on the y-axis is the change in oxygen saturation pre and post PDT, and towards the arrow mark that you show here is the increased oxygen saturation post therapy. So if your dose is not appropriate or is not sufficient to actually shut down all of the vessels, you can actually increase the oxygen saturation as well. And based on that, you can actually see a few tumors that had increased oxygen saturation had greater rate of volume increase compared to tumors that had less oxygen saturation. These are very few mice. We are only seeing trends, but we have to build a lot more to actually make sure that a lot more mice to make sure that this trend actually holds. So where are we going next with all of this is we want to build a multi-parametric treatment prediction map from this. Ultrasound is great at providing tumor volume, necrotic regions, also blood flow, Doppler, power Doppler, I'm sure all of you are aware of that. And photoacoustics can give us the vascular density, oxygen saturation, avascular regions, and we are trying to collaborate with the Data Intensive Studies Center at Tufts to build a label-free multi-parametric treatment prediction maps. And I just showed you one example of using change in oxygen saturation that can actually predict treatment response. All right. So we'll go on to the next topic here on planning treatment combinations. Again, talking about tumor hypoxia, that's again a big problem, not only for PDT, but also for radiotherapy. So several researchers have tried out different ways or methodologies to alleviate tumor hypoxia. Some of them is like using, vascular normalization agents, bevacizumab, or, trying to do some stromal reprogramming. A lot of, drugs that are out there that are, that show that tumor hypoxia can be alleviated, can be combined with radiotherapy to reduce the hypoxia and make these therapies much more effective. So since we can image, oxygen saturation with photoacoustics, one of our thoughts was, can we time that combination treatments, right? When do we give the chemo drug? When do we give the PDT drug? And can we time it accordingly so that the outcomes are much more impactful, right? Can we impact the cancer treatment outcomes? So there are a lot of FDA-approved drugs. We are particularly focused on Onivyde here, and I'm going to show an example from Onivyde, which is approved recently for pancreatic cancer. And in orthotopic tumors, it's shown that it reduces hypoxia. This is an immunofluorescence stain that is for HIF1. You see a reduction in HIF1 alpha intensity 24 hours post this Irinotecan treatment. We image the mice also with photoacoustics, and you can actually here see decrease in HIF1 means increase in oxygenation, right? So this is opposite to the PDT story that I was talking to you about. So before treatment, again, the tumors are very heterogeneous. You see a lot of blood vessels here, a lot of hypoxia. But post-Irinotecan treatment, you see an increase in oxygenation. So because the oxygenation is increased, now you can sort of do PDT when there is more oxygen in the environment, and PDT will be much more effective. So we call this photochemotherapy, and the idea is that you have your chemo drug. In this case, it is Irinotecan. It does DNA damage, and because of that, there is a reduction in tumor hypoxia, increase in tumor oxygenation, which is going to help the PDT. So if we try to target or do our PDT right at that 24-hour time point, when tumor oxygenation is high, the hypothesis is that you would get more effective outcomes. So here you're seeing some pancreatic tumors that we treated with PDT, which is shown in cyan group. Irinotecan shown in green. Obviously, they were not that effective in this particular cancer cell line, but photochemotherapy, you do see a reduction in tumor volume, and you can actually see, compared to other treatments, the tumor volumes in photochemotherapy is much smaller. We also did H&E and actually found that the necrotic volumes in photochemotherapies are much higher compared to the no treatment or the single treatments alone. Now, we tried the 24-hour time point, but obviously we also want to do more on this work. Should we target these tumors at multiple time points, and can this tumor oxygenation imaging help us in trying to identify or time the PDT and also the chemotherapy accordingly? Moving on to the third part, which is monitoring the nano responses. Again, going back to PDT again. So light, the photosensitizer, and oxygen. These three parts are very important for PDT to be effective. So we need to be able to deliver photosensitizer specifically to the tumors, and also there needs to be oxygen. Right? I showed you one way in the case of Irinotecan, it is alleviating tumor hypoxia. But are there other ways to actually do that? Yes, there are some hypoxia-activated prodrugs, and what we try to do, as other groups as well have tried, is to deliver oxygen through perfluorocarbon nanodroplets. And it is very important, again, to alleviate hypoxia for PDT, because hypoxic tumors show decreased PDT response. So if you have very hypoxic tumors, PDT response is not that great. More oxygenated tumors, the PDT response is good. So we need to be able to deliver the photosensitizer and oxygen, and can we do that all together in one nanoparticle? Right? And to do that, what we have here is a nanodroplet. So basically, it's a perfluoropentane liquid core that's carrying oxygen. And we have the BPD, or the photosensitizer. BPD stands for benzoporphyrin derivative. It's the chemical that we use as a photosensitizer. And we also use something called indocyanine dye, ICG dye, to actually monitor where these nanoparticles are going in the tumor. It is a very good photoacoustic contrast agent as well. So BPD is used for photodynamic therapy. ICG can be used for monitoring photoacoustically where these nanoparticles are within the tumor, and also photoacoustic imaging can be used to monitor the oxygen delivery. So I'm going to show you a couple of images here before and after injection of the nanodroplets. So we have the ultrasound images here of the tumor. This is before injection. Again, red is oxygenated areas. This particular tumor is very hypoxic. You can actually see a lot of blue regions here. After injection of our nanodroplets, you can actually see an increase in oxygenation in these tumors. To increase, you see all the increase in the red signal. These nanodroplets are delivered through the blood vessels. So you can actually appreciate it's much more sporadic. That means the nanoparticles are going through the blood vessels and just oxygenating around that regions. We looked at the PO2 values, which is a partial pressure of oxygen electrode. But again, this is a point source, right? It's not imaging the whole tumor there. You're just looking at a particular location in the tumor. We do see a significant increase in the PO2 values of the tumor using a standard oxygen electrode. And what we actually notice is once, for several mice, when you do the statistics, is the histogram of oxygen saturation is switching or moving towards oxygenated tumors. So red is before injection, green is after injection, so you can actually see, appreciate the movement or the histogram moving towards the highly oxygenated areas. The total hemoglobin remains the same because total hemoglobin is representative of the vasculature, right? By delivering nanoparticles, we are not doing anything to the blood vessels. So that's like almost like a control for us. And within this oxygen saturation, we try to decipher what is happening to highly oxygenated or larger blood vessels versus hypoxic regions. So this oxygen increase is actually happening in low StO2 areas. So wherever there was low StO2 or low oxygenation, we increase the oxygenation using these nanodroplets. But in high oxygenated areas, places where there's already enough oxygen, the impact was not great. And obviously, the oxygen sensor does show a significant increase in the partial pressure of oxygen. We also did histology to actually confirm that we are reducing hypoxia. This is a control image or a control tumor where we stained for hypoxia shown in green through the pimenidazole stain. The red is the blood vessels and the blue is stained DAPI. These two inserts are shown here. You see a lot of hypoxia in control tumors. It is sort of expected. It's very heterogeneous as well. Even surrounding some blood vessels, you actually see a lot of hypoxia, which shows that these blood vessels were dysfunctional. And in the case or in the mice where we injected the nanodroplets, right, we don't see as much hypoxia at all. And when we try to quantify that for several mice, the amount of hypoxic area for our nanodroplets significantly decreases. So that's great. So we are delivering oxygen, we are delivering the photosensitizer to the tumor. So now if we do the PDT, it should be very effective, right? So that's what we try to do here, evaluate the in vivo PDT efficacy. So this is a tumor volume showing days post-treatment. The mice were treated on day 0. No treatment mice, obviously the tumor is continuing to grow. And Visudyne is the clinical formulation of photosensitizer that's out there. Is not as effective because again, if the tumors are hypoxic, PDT is not that effective. So you can actually see tumor volume is not reduced as much. But the nanodroplets or nanodroplet enhanced PDT, we do see a reduction in tumor volume significantly. And also there's a lot more necrotic zone in these tumors. So in summary, I've shown you, the basics of ultrasound for acoustic imaging, the PDT, how it's being used in clinic. But again, the roadblocks are hypoxia. And if we can alleviate hypoxia, PDT can make a huge impact in clinic as well. And ultrasound-guided photoacoustic imaging can help, achieve that by stratifying subjects from responders to non-responders. And from the responders, we can also able to predict the tumor recurrence, right? We can aid in designing combination therapies. I showed you an example of Irinotecan and photodynamic therapy, and then monitoring therapeutic response. If you have nanoparticles that are delivering drugs or oxygen in our case, we can use photoacoustics to also actually monitor that. The current ongoing studies is we want to, again, determine the timing in photochemotherapy and also understand what exactly, is happening with these prediction models. Can we make them more robust? What is the sensitivity, specificity? A lot more needs to be done. This is all very early work. And another part of my lab also works in building these photoacoustic systems, handheld probes, which I have not shown today, but build clinically translatable photoacoustic imaging based dosimeter tools that can be used in clinic for, say, oral cancer specifically. Some of this work is reviewed recently in a publication from our lab that talks about role of ultrasound and photoacoustic imaging in photodynamic therapy. So all of the things that I talked about are on there. And with that, I would like to acknowledge the IBIT team, the graduate students and the undergraduate students, Marvin Xavier Sullivan specifically, who worked on the nanodroplets work, and my mentors and collaborators at Tufts and also MGH and Nanohybrids Inc., who we collaborate on the nanodroplets project as well, and funding from NIH. Thank you.
Thank you so much.
Thank you.
Before we get into the question and answer session, I just want to call everyone's attention to our next hybrid seminar, which will take place on Friday, April 15th next week. We will be hearing from Professor Brian Bryson at MIT as he presents engineering host-pathogen interface. Also, we want to remind you that on April 23rd and 24th, Columbia's Biomedical Engineering Department is hosting the 48th Annual Northeast Bioengineering Conference, also known as NEBEC, N-E-B-E-C, here in New York City on our Morningside campus. Registration is open until April 13th, and you can go to, is the website on there? nebc.org, which is in the bottom right-hand corner of the screen there. Please follow us on social media to receive the latest updates. And you can always access registration information on all of our seminars on the Biomedical Engineering Department's website, bme.columbia.edu. Okay, and then we have the question and answer slide here. So we're going to do a combination of questions from people who are online as well as people in the audience. Once I get the microphone. Okay. Questions? Yeah.
Okay, good. I really enjoyed the talk. I have a couple of questions, but I'll start. So it seems like the tumors that can be treated this way have to be pretty superficial. Is that correct?
Yeah, so the question is, do tumors that are undergoing photodynamic therapy, do they have to be superficial? Yes, and also no. Yes, if you're directly shining light on these tumors, yes, they have to be superficial because light penetration can be an issue. But now there is all the technology of fibers and implantable fibers. So you could also do endoscopically, though that's still a lot of research is going on how to do deep tumor photodynamic therapy. There is a possibility to do that as well by using fiber proteins.
And then another sort of different question. So the imaging part was very neat that you showed in the beginning. Is this whole technique used in altogether different ways as well, or is it mainly used for like cancer?
It is used in several applications for developmental biology, looking at hypoxia, say in placenta, rheumatoid arthritis, and a lot of other things as well. It's not dedicated to cancer. So any location or any where there's oxygen saturation change that you can image photoacoustics will be useful. And not only that, you could also inject contrast agents that are targeted towards a particular disease, and you can image those contrast agents using photoacoustics as well.
So the photosensitizer that you put in is particular for this oxygen saturation. Okay.
Yeah, for PDT, for photodynamic therapy.
Yeah, great, thanks.
We got a we had a question from Murad Hussein online. He said, nice talk. What kind of light do you use in PDT? Is it localized like laser in photoacoustic imaging? And have you tried to use this method in treating tumors at later time points? And finally, how does tumor size impact the treatments?
Yeah, so there are three questions here. The first one is what wavelength we use. So the benzoporphyrin derivative photosensitizer that we use absorbs at 690 nanometers. So we use that red light, 690 nanometers, to do photodynamic therapy. The second question is it localized like in photoacoustic imaging? Yes. So we have a laser that points, through an objective points on the tumor. So it is a localized therapy, and that's the beauty of photodynamic therapy is because it's spatially localized, unlike in radiotherapy where you can have a lot of systemic effects. And the third question is, have we tried treating tumors at later time points, and what is the size impact? So if you keep the dose of the PDT photodynamic therapy constant, and you have larger tumors versus smaller tumors, obviously smaller tumors are going to respond well compared to the larger tumors. Yes, so there is going to be a size dependence. There's also a publication on that front that we actually did a sort of correlation analysis using size dependence and PDT efficacy. But again, if you use different methodologies, or if you use fractionated treatments, or you treat the tumors, larger tumors at multiple time points instead of using a single PDT dose, you will be able to treat them well.
I have a question. You said in a bunch of spots that you made a point of there being no contrast agents. But what if you have contrast agents? Are there all kinds of special tricks you could do that would make things even better?
Yes, absolutely. And the no contrast agent point was coming from the clinical translation aspect of things. But if you use contrast agents, say gold nanoparticles have very good optical absorption and are very good for acoustic contrast agents, but then they have biocompatibility issues, right? There are some dyes, for example, indocyanine green is already FDA approved, but if you can tag that along with, say, any biomarker, say, that's overexpressed in cancer, then you can inject that into the body and actually identify the tumor margins along with oxygen saturation. Because this is wavelength dependent, you can do multi-wavelength imaging to tease out what part of tumor has nanoparticle or dye or contrast agent accumulation and what is the oxygen saturation. You can get that information simultaneously.
And I had a couple questions about, so you showed that the tumor data was very clear when the tumors responded or did not respond. But maybe like an interesting case, that one in the middle you talked about, but I think you labeled it regrowth, right? And I had two questions about that regrowth condition. Number one, the plot in the top of it, it looked like there was a very sharp line between the two. And that almost seemed like too clean to me. Is that real?
So as I said, we used a threshold algorithm, right? So when you use threshold algorithms, those lines are clean. That was our first pass at trying to build the prediction models. But as we move on, it's not going to be as clean. It will be more pixel based, right?
Okay.
So it's more for representation.
The raw data are a little messier than that.
Yes,
It doesn't
yes.
matter for the person investigation.
Yes, yeah. So because I use a threshold for the classification, the line was clear, and it's more for the representation purposes that I showed it that way. But we have the whole thing.
That's fine. And then how often does that recurrence case happen?
In this case, about 40% of the time. If we
Okay,
don't treat
so
the
it's a big
tumor,
deal.
it's a big deal.
Big deal.
In all therapies, not only in photodynamic therapy, but I think in radio, chemo, also recurrences is a big deal.
There's one more question.
Yeah. We had a question from Pavan Setbakti online. Thank you so much for the talk. How do you decide which type of photosensitizer to use? What type of photosensitizer?
Yeah, thank you. So there are several types of photosensitizers out there. We chose to use benzoporphyrin derivative because it has a very good singlet oxygen yield, and photosensitizers are qualified based on how much of singlet oxygen they can generate. If a particular dye molecule is a very good absorber, probably is not a good photodynamic agent. So benzoporphyrin derivative has very good singlet oxygen quantum yield. And the second aspect that we use to decide a photosensitizer is also the wavelength at which it absorbs. You want something that absorbs in the near infrared or close to the near infrared. Benzoporphyrin derivative absorbs at 690 nanometers, which has higher deeper penetration depth than something like protoporphyrin 9 or ALA, which is in 635. That's another commonly used photosensitizer out there in clinic.
Are there any more questions here? All right, well, thank you so much. That was wonderful.
Thank you, and thank you for the invitation.